a topic that I personally am very excited about because I'm going to show you a nice online tool to handle this type of data, DNA microarrays, the data that comes from these microarrays. DNA microarrays is one of the very powerful technologies that help us in measuring gene expressions, for example, for different conditions and experiments. So it was invented in the late 1980s and then became widely used in 1990s. Nowadays, the trend is to use what's called next generation sequencing, RNA-seq, things we're going to learn more about in the future. But certainly DNA microarrays is this very powerful technology that helps to study and measure the expression of many genes at once. Not few genes, many genes. The idea depends on placing thousands of genes in known locations in a solid surface, okay? And by now you understand fluorescence and fluorophores, these concepts of our toy that we eliminate light to, and then could be the laser through these technologies. It is in fact lasers, and then based on the emission of the light that we see then, and we are able to measure, we're going to be able to tell if this is given, you know, greenish, reddish, yellowish, what kind of difference is we are being told. So DNA microarrays are usually kind of glass or in terms of material or silicon. DNA are attached in a kind of predetermined grid fashion, as you see here. So each spot, before I come to here, the idea is think about this chip. Okay, we have kind of a chip, and each spot will represent a probe. By now, we probably know what is a probe. So let me ask you this question. We have now accumulation of many terms, but let's have this as a treasure hunt question. What is a probe? Just, you know, write down whatever you think. What is a probe? Okay. A probe is this. Let me give you a moment to think. Okay. So a probe is a piece of the DNA. Usually it represents a single gene. So we're interested to, you know, have one of the areas here to be relevant to maybe gene A, and maybe this is gene B, and maybe this is gene C, and we keep going. And based on these colors, we kind of tell, okay, this is gene B, this is more reddish. So if we have two experiments, and we know this is after the treatment condition, and if high intensity of color, higher intensity, which is high level of greens, would mean for us something higher than this gene is perhaps upregulated. The grayish ones or yellowish ones may mean neutral. When we come to the green one, maybe this is gene X, this is downregulated. So these terms of upregulation or downregulations means that that gene, in terms of an expression that comes all the way from mRNA as a sample prepared in the lab, may indicate that this gene is more active as compared to the other genes. Now, is that something normal, disease-related, specific exposure related? This is subject to further analysis. The starting point in terms of a technology, there are certainly lab technicians working on preparing the samples. We can imagine as we speak these ideas that there is an RNA extraction. It is gene expression measurement. So we are looking at mRNA extraction, cDNA, of course, and then the probe designed to have what? Some hybridization by the end of the day. So what is hybridization, which is the principle of DNA microarrays, okay? let me remind you about some very interesting concepts which we discussed before. Through two hydrogen bonds, we know that adenine links always to thymine. And so through three hydrogen bonds, we know that cytosine always binds to guanine. So with the right conditions and given this information that A's always binds to T and G always binds to C or vice and vice versa. By knowing this, can we take advantage of this information? And we have been seeing this kind of concept before in terms of, given or preparing the right conditions and environment to have some interaction. Things about polymerase chain reaction, DNA replication, so on and so forth, all of these ideas. Now, in terms of a DNA microarray, the probe that is designed specifically to hybridize to specific target of DNA sequence. So we are interested in knowing DNA sequence. Basically, we're talking about cDNA sequence because this is the inverse of mRNA, sort of. So we are trying to measure the quantity of that. We're not doing here, we're not trying to read the DNA. We are trying to measure the quantity of expression. Hybridization, where the labeled target of DNA is going to be presented with certain conditions. So the DNA is going to be washed over the microarray slide. So each will of those, or each position will, will is not the right word here. So don't be too sensitive to these terms. If you are lab technicians, let's try to use some terms, to try to bring you closer the idea. Will would link more to PCR maybe. And anyway, let's say this is a spot here for that specific gene. So there is a probe here. There is a probe that is designed. Okay, and that links, that probe comes from the specific gene. So we design those. Okay, it could be the company, scientist, whatever. And then DNA comes from some sample. Okay, some sample. We have here some sample, and we have some DNA, or again, cDNA coming from RNA. And then we drop this in this whole microarray, which is kind of being washed over the microarray slide. Now, if the target DNA contains sequences that are complementary to these probes, then it's going to bind to it. And based on that binding and hybridization, what's going to happen? Tell me, okay, what's going to happen now? Once there is a hybridization, another treasure hunt question is, what do you think is going to happen? What has been the protocol or the standard or idea so far in many of the discussions? If we design something, this is not happening in our cells, so we purposely add some sort of fluorophore. So some fluorescence light is going to come out that would be detected by some machine. Okay. So as an example, imagine we have this like 5' direction, and then we have A, T, G, C until we get to the 3'. And what is going to be now this kind of a probe, if this is a probe for us, it's on the chip, okay, and then we get the DNA sequence. In this case, what's going to be that? It's going to be 5', okay, and they will have T, A, C, G to 3', okay, in terms of a directionality, and then they both are gonna hybridize, and this is gonna be a target sequence coming from DNA. So source of this is our cDNA library, and when I say DNA for short, this is what I mean. Okay, and this is the probe that is designed, and this is already sitting in its own house. Imagine this is a piece of land, and there is a nice house here, and the visitor is going to be this sample. Is going to knock on the door, and if we can bind to each other, let's make some celebration, send some fires around, fireworks, and this would mean, oh, there is a strong binding, okay. that is DNA microarray. There are different types of DNA microarrays. There are gene expression microarrays. Okay, help us to know the gene expression of microarrays. There's what's called SNP microarrays. So these work with in a different way. And there's what's called tiling arrays. Now, these are three different types. The idea, in principle, it is, you can think of the same technology, but what is different is how probes are being designed. How probes are being designed. We can tell that if we're working with gene expression, the probes are going to correspond to known genes. Makes sense, right? If I'm going to measure something and I want to know its expression, I need to link this to some gene. If I'm going to look into SNP microarray, which is single nucleotide polymorphism microarray, this is meant to work on some SNP locations in the genome, more trying to define, more of looking at trying to link things into phenotypic aspect. So these are more kind of probes that are allele-specific oligonucleotides to detect the polymorphism within a population. So the probe design is going to be different, okay? So it is linked to allele, not a gene. And for the tiling arrays, it's more about genomic area of interest. In that case, what we're looking at is the probe is going to be overlapping, okay, types of probes across the specific regions of the DNA, okay? So in this case, this allows, the way you think of it is we don't know specific reference or gene. We are interested in novel sequences, and this is one of the ways if we're going to detect unknown or uncharacterized sequences. So there are different types of DNA microarrays. In general, there are two approaches to work with array platforms. There's what's called dual channel labeling, and this use different dyes like CY3, which is for fluorescence green, and CY5, which is for fluorescence red. So this kind of microarray platform can deal with two colors, which means two samples at the same time. So this can be used for two samples. Another single channel technology, which means it is single color, is going to, of course, have fluorescence. Fluorescence is always there. But again, only one type of fluorescence or one color of a fluorescence, a green, for example. And in that case, this can be used for one sample. Okay. And, while a dual channel can allow for direct comparison of two samples on the same microarray slides, this can reduce variability, for example, and improve data accuracy. Single channel itself can be helpful for performing more consistent large-scale studies when more samples need, for example, to be analyzed. For example, also doing simplified data analysis, since you have one fluorescence intensity for each single gene. So, the choice between these, it's one of the things that choosing which method depends on the specific research question and experimental designs. Dual channels allows for simultaneous comparison of two samples on a single microarray. Single channel analyze one sample per microarray slide, and which provides, of course, absolute expression data. So in terms of how dual channel is going to work, we have samples like control and experimental. Think about building or designing certain experiment and having some treatment conditions. Of course, there is some control because we're going to compare the expression across these. So we can have a fluorescence attached to it, like one of the dyes. In terms of the library as we prepare these samples here, or this cDNA, And then we have the other one labeled with different colors. And then we kind of subject them onto or wash the samples at the same time through the same chip. That's going to be go through some laser emissions that eventually will give us some output. Okay, the color in this output in terms of intensity levels is going to indicate some meaning. We know each circle here indicate one specific gene. So this could be upregulated, downregulated, or maybe, you know, there's no specific realization of the expression. Let's watch this interesting okay, video. Okay.
This animation will demonstrate how DNA microarray experiments are performed. DNA microarrays, sometimes called DNA chips, reveal the expression of thousands of genes on a solid surface, such as a microscope slide. In this example, we'll use yeast as a model system to illustrate one use of microarrays. One common use of microarrays is to determine which genes are activated and which are repressed when two populations of cells are compared. Every gene is measured simultaneously. As an example, we'll compare what happens to yeast genes when cells are grown in aerobic versus anaerobic conditions. The cells grow and adjust which genes need to be activated or repressed in order to survive. Now it is time to isolate the mRNA from both populations of cells. The cells are spun in a centrifuge. Now that the cells have gathered in pellets, we remove the liquid, but not the cells. Next, it is time to extract the mRNA from the cells. When we add the extraction buffer, the mRNA is released into the solution. Next, we remove the RNA and place it in a fresh tube. Now, let's make the cDNA from the mRNA. Here we see 3 of the many mRNA molecules from each tube of cells. Each mRNA is converted into red or green colored cDNA. When the colored cDNA is made, the mRNA degrades. Then we combine the red and green cDNA, mixing both colors into a single tube. At last, it's time to look at the DNA microarray. In our experiment, a microarray or DNA chip contains about 6,000 spots. Each spot is a different yeast coding sequence from a different gene. Let's choose 3 spots at random to follow in detail. Each spot is made of DNA that can base pair with its complementary cDNA. Here are partial sequences from each of the 3 spots we are observing. Now, let's incubate the mixed cDNA with the DNA chip. For the sake of our example, we'll zoom in and show that some of the labeled cDNA have bound to the DNA in the spots and formed base pairings. Here we see green and red cDNA bound to this spot. Only red cDNA is bound to this spot. And only green cDNA bound to this other spot. In a real experiment, you would not see any of this detail. You would only see the original microarray. Now we must wash off the unbound cDNA to see what is bound to the microarray. Let's detect the bound cDNA so it can be visualized. We begin by placing the microscope slide containing the microarray inside a scanner. We'll examine the next phase of the process, keeping our focus on the 3 spots we've been following. First, a green laser scans the microarray. The resulting image is stored on a computer for later analysis. Now it's time for the red laser. This image is also stored on a computer for later analysis. Now, we move to the analysis phase. After we eject and safely store the microscope slide, we retrieve the red and green images from the computer and create a merged visualization. In the merged image, we see an aerobic gene labeled in green, an anaerobic gene labeled in red, and a gene labeled in yellow that was expressed in both aerobic and anaerobic conditions. This is one example of how DNA microarrays are used. In an actual experiment, quantitative analysis would be conducted on all 6,000 genes.
Okay, this is an interesting animation. And again, the output of this process is going to be a set of images that we will feed to what? To some computer program. And you can see here some sample images, right? You can tell there are some issues with some of these images. In fact, if you review the process, you can kind of, that there can be some issues as these lasers are getting scanned. It's kind of technology limitation, which is generally expected in everything we are facing with these technologies. And it is part of the process. So the variation in DNA probe concentrations, the one that we look at here, you can see this is big, small, can significantly impact the quality and the DNA microarray results. So there are many techniques that one can look into. For example, looking into the probe in terms of biotechnology aspect, make sure that all the probes on the microarray have a similar concentration, array printing quality, look into sample quality, hybridization conditions. As bioinformaticians, we focus more on the data analysis aspect. So we try to perform some specific kind of normalization. It's always the case. We have to examine the quality of the samples and then start to see if we can actually go and perform some normalization, proper heterogeneization methods to correct for this systematic bias, okay? For example, the variations in the probe concentration or labeling efficiency and so on and so forth. Also, it could be that one of the techniques, once we are done, why we don't move to qPCR, okay, which something we introduced before polymerase chain reaction. It is the golden standard. That is the one which is very accurate, but we can run it for maybe a limited number of genes, so we can go there and then do validation and so on and so forth. So one of the known technologies out there for DNA microarrays is the one by Affymetrix. Affymetrix gene chip platform is one of the pioneering and most recognized technologies. This is an image here that illustrates that gene chip. So this is kind of zooming in into this 1.28 centimeters. And at the micro level, you can see one spot how many probes it's going to have. Okay, there will be a group of probes and copies that we call oligonucleotides because they are short parts of probes, short sequences. Okay, and this gives you an idea that there are thousands, okay, of different complementary probes are designed by the end of the day to what? To measure the gene expression of roughly 20,000 genes plus. Affymetrix microarrays use what's called photolithographic synthesis. Okay, photolithographic. Okay, photolithographic synthesis, and this is this is kind of technology that brings the design of probes similar to, the industry of semiconductors and semiconductor manufacturing, if you ever hear about it, for our chips and CPUs. So this is that kind of, this is another technology here, but in a similar fashion, it involves synthesizing DNA probes directly on the chip, okay, using this whatever the term here, photolithographic, which would refer to something like light-directed chemical synthesis. It helps in designing high-density arrays. So there are millions of copies of specific oligonucleotide, and these copies are there. So there are applications of these copies to help in measuring the gene expression through the hybridization principle and so on and so forth. Part of the design of the Affymetrix technique is what's called a perfect match probe and a mismatch probe. So think about this sequence here. Let me write it down. ACG, TTA, for example, C. Okay. Now this is a perfect match designed probe, which means once the DNA target sequence is going to bind to it, a fluorescence is going to come and give us an indication that there's this binding. Now, there is what's Affymetrix did this design. So they said we'll have ACG, okay, and here we're going to make a change in the middle, TAC, okay, one single mismatch here in the middle. So you can think of replacing this with G, for example, only here, okay, something in the middle in general. That is what's a mismatch. So why they would have a mismatch? Why do you think there is a need for a mismatch? So I'm going to ask you this question, and you can share with me your answer for this question. So mismatch probe helps in estimating the background noise in the microarray experiment. The noise can come from because of different reasons, from various sources. Could be like something like a scanner noise, okay, or maybe a fluorescence from the substrate. So the process of hybridization in general is not perfect. It is imperfect because someone can come to and say, can come and ask, well, we know that this is going to bind to a target sequence. Okay, so this is going to bind to TGCAA. TG. Okay, we know that this binds to this one, right? Now, is there a chance that this binds to this one? Yes, even though it is weak, even though it's not very stable, and even though it's non-specific binding, this could happen, okay? So therefore, this idea of a mismatch is kind of a sanity check into what is happening in the background. So we can do some correction. It could be, again, technology aspect. Something went wrong in terms of the experiments, the hybridization process itself. There could be different reasons that hybridization is imperfect. There could happen non-specific binding, something called cross-hybridization, where the genome has closely related genes or repetitive sequences, and the probe that is designed for one gene might still bind fragments from another gene. So this could happen. So these are the two platforms again. The Affymetrix gene chip, generally it deals with one single chip. So these are the steps. There is going to be a labeling of the RNA. Eventually we will get to have this over the chip, and then do the preparation, try to scan the colors. Okay, this is the flow for a gene chip. It's going to go through these steps of RNA extraction, of course, cDNA preparation. There will be a fragmentation step. Hybridization is very important at this stage when you put the cDNA samples. Here, what you see the difference is on the left side, we are starting very similar to here, but although there are some differences, but in general, what we have is two groups or two samples, each representing a different condition. So with cDNA arrays, since we'll have images with the red scan of a laser, we'll have R values. Think about RGB values. We have red intensities representing the red color, and then the green values. The ratio of that is going to indicate for us, okay, the level of the change. We can apply log here and as a way to normalize this, okay, and try to see in general the differences in terms of upregulated, down or down regulated, or down regulated. Yes. Affymetrix on the other side, it handles this differently because we know that we have a group of perfect matches, some mismatches. So we will measure. This is going to be, was designed to have it act as a sanity for the background noise. So we can take this mismatch, subtract it from the perfect match, and as a quantification of what is going to be eventually a reflection on the level of expressions of that specific gene, okay? So microarray data analysis goes through different steps, starting with pre-processing. There is a quality checking that needs to be done. Try to visualize the data, see if there is any issues with the whole process. There are two aspects for that. It's for the biotechnician through the laboratory setup. And this is also the bioinformatician who's they work closely. We are an interdisciplinary field. Even I'm gonna do data normalization, perform statistical analysis, give the list of differentially expressed genes, apply some clustering, and then show interpretation for the results. I'm going to communicate this to the whole team. I've went through the steps a little bit quick because I'm going to illustrate them through a demo very closely. Microarray data analysis, we can look into, always it's highly recommended that you have 3 or more biological replicates because you're going to perform a statistical analysis. It is some kind of, sometimes, you know, when you deal with some experts, they share the insight that's very difficult to run one simple experiment or costly, difficult to run one experiment. So it cannot be difficult and simple at the same time. But again, it's simple for me as a bioinformatician, difficult for them. So that's the confusion. Anyway, so it's going to be costly. And then as a bioinformatician, I need more samples. So it's very important if you are working in your lab or gonna involve teams to have discussions right from the beginning. If the whole study ends up working really on, you know, just generating one sample from treatment group and then one sample from control group, although it is tedious, taking lots of resources and time, that is not gonna provide any significant insights or even meaningful results. So there are biological replicates, so take this into consideration in terms of designing the experiments. Work closely with the experts from the different domains. Technical replicates, this is more for the industry and the technology aspect. This is one of the interesting figures that are common to see the gene expression data that I've just introduced coming from the microarray. So we'll have rows indicating genes, columns indicating samples, and then kind of the clustering happening at the row levels and the column levels. Using some distance metrics is going to indicate for us color intensities. So we can tell, first of all, how that samples group together in terms of, you know, looking at the annotation, are they in the high dose or medium dose? And then looking at the gene as well. Some genes are showing differences between two groups. Some genes are not showing significant changes. So maybe we are going to focus more on these genes in our experiment. So let me take you to one demo, and I hope you will enjoy that one, and then we'll finish up with it. So what I'm going to take you to here is a website. Oops. Okay, this is called Network Analyst. It's one of the known websites for comprehensive gene expression analysis. I've contributed to version 3, Network Analyst 3 in 2019. However, this was developed early by Professor Chiongwea Jefsia many years ago. So if I come here and click on gene expression table, I can choose and to try some of the examples. What I'm going to do, I want to share my whole screen. I'm just worried if there are some pop-ups that you can't see them. So maybe then this is going to be better. So in terms of try examples, you can have some options here for the species and different options. Now let's try an example. So this is one example for a microarray data of 30 samples showing different conditions and exposures of primobenazine on rat liver. So this is more of a toxicogenomics based study. And this is how the data looks like. Of course, this is after handling the raw data files. When the experiments are done, each sample is going to have full file. Once this is processed, we'll end up with the following: a very nice looking data set. This is kind of a matrix, which means we have rows and columns. Rows in this case are indicating some IDs, like this ID here. This ID means something in some database, which probably would link to a specific gene name. And then we have columns as samples. And then we have also annotations, like what doses we have for each sample. Okay, so you have replicates. We know for the control group, we have 5 replicates. For the low dose, we have 5 replicates, which is nice. That seems an interesting data set. So I'm going to click submit. I'm going to use this sample data set for purpose of illustrations. Now I'm going to skim or quickly go through some of the steps. I'm not going to dive so much into this, but feel free, if you're interested, to ask more questions, to come back to me. If you have a data set at your lab, or if you're interested to work with this, also we can discuss it. So this is going to start with giving me a summary, like how many features being. At this stage, we may be looking at, we're talking about props, so some prop IDs. So how many prop IDs linked to genes, okay? So we have that number. Still, some of them were unmatched. Now, these are the data. We see, as we look into the different samples, we see some variations. This is because of the technology. It shouldn't be expected because all samples in general are going to carry similar level of information. We have genes. Roughly how many genes? We have almost 14,000 genes. So each single sample is going to report results in 14,000 genes. Now, while for one specific gene there might be differences, but overall as a distribution across samples, we need to expect there is some similarity in terms of median. The source of variation here is telling me something about the technical aspect that we have just raised. Could be some noise that we need to fix, okay? Like similarly, this density plot is showing me some variations that we don't like to see in terms of quality check. So what do? Easily we can come and then we can remove some of the, outliers. You want to call them outliers, noisy data. We apply some well-known normalization techniques like quantile normalization. So I'm going to submit this one. And here you go. Now, in terms of technology, so I don't want you to confuse this with the genes. This is for the samples as a distribution across samples. For the genes, yes, we want to see differences, okay? Otherwise, what's the point of the experiment? So I move ahead and we can use some statistical analysis, which we refer to as differential expression analysis. Under certain conditions, we have some p-values to try to judge the significance of difference between two distributions of gene scores, something that we'll probably also dive into in the future. But for the time being, there are some methods. One of the common methods for microarray data is lemma. Now, here where we start the cool part on the next page. So what do we see here? We see a summary of information. If I click on this visualization here, wow, so I find there is a gene, and across different conditions, there are significant changes. So this seems the highest top gene here in terms of p-values. So we have access to it on NCBI database. This is one of the known databases to reference and document the genes. So this is ME1, okay, gene. And again, it's related to the rat. And then we have full information here. So given my own study, I know that this is the gene that is, highly responsive in that case. Okay, this enables NADP binding activity and malate dehydrogenase activity involved in response to hormone. Okay, and so we start trying to interpret the results and go through this information. We have other list of genes here that we can go through, and if I keep going, let me go here. There will be probably some list of insignificant genes, okay? Genes that will show no variation between the different conditions. In this case, maybe you are not interested to look into those, okay? Let me choose maybe something here. There is some variation, but not as significant as the first group. So I'm going to proceed here. Now, this is, I guess, not going to work with that number of genes. So I'll just go back. This is what's called benchmark dose. Let me fix this here, or just add some condition. Say, look, twofold change. So 200, we'll end up with 235 significant genes instead of 2,000, which were a lot. Now we apply this kind of polynomial fitting on the gene expression levels, and then it's going to be an aggregate analysis to tell us, given that we have a set of benchmark doses, what if we try to aggregate these insights on gene expression level of all genes and try to derive what could be a safe dose. This could be helpful in knowing, giving a chemical, what is the dose that should be safely given for a specific treatment. Okay. I'm gonna here where you have set of tools that you can play with, like heat maps just discussed and social networks. So I'll jump into volcano plot, for example, which is one of the standard tools to visualize the gene expression information coming from these data sets. So as we do the analysis, it's going to be very interesting if we can visualize that for easier exploration. This shows the x-axis, the fold change, log fold change. Usually looking for higher fold change, okay, or maybe drop down fold change. So is it going to be a much down regulation, significant down regulation? And we're also going to look at the p-value. So we look at these two scores. So given the symmetry here, we end up with what's called the volcano plot. And we are interested in learning more about some of the genes. So we can click here and see the information and go back and see the distribution. So I hope that exposed you to nice tools, and that you enjoyed this discussion for this module. See you with the next one.